So the next speaker will be Camille Coustaury, a second-year master degree student in engineering at the Institut Polytechnique de Paris.
Yes, thank you. I will just share my screen. Can you see my slides?
Yes.
Okay. So thank you very much for having me here today. So as you said, I'm currently a student a master student in engineering and statistics for health science at Institut Polytechnique de Paris. So this summer I did an internship at CHU Saint-Justine, supervised by Benjamin Deleneur. And so I worked on a project which is a collaboration between Neuropoly and Neuro. So today I will present you the project on spinal cord quantitative MRI and how it is sensitive to microstructural changes in patients with Parkinson disease. So first I will make a quick introduction on Parkinson disease and the motivation of this project. So as you may know, Parkinson disease is the most common neurodegenerative movement disorder with a prevalence of 1% in the 60+ age group. So in Parkinson disease, there is a progressive death of dopaminergic neurons, and these neurons are involved in the movement control. So this induces some movement disorders such as rigidity, slowness in movement, or tremors. There is also some additional burden to disability. In fact, with Parkinson disease, there is an increase of depression and anxiety prevalence for the patients and for the caregivers. So the motivation of this project was that there has been shown in previous studies that Parkinson disease has an effect on spinal cord, but no previous studies had been made on the sensitivity of spinal cord quantitative MRI to the microstructural changes in patients with Parkinson disease. So our main question was to find if we could find some potential biomarkers of Parkinson disease in the spinal cord. So next I will present you the data we used and how we processed the data so we could analyze them and show the results. So we acquired several types of MRI of spinal cord. So T1 weighted images, T2 star weighted images, and magnetization transfer images, so that we could compute the magnetization transfer ratio. So all these images were acquired at cervical level. And for the T2 star weighted and the magnetization transfer images, images were acquired with slice at the same physical position, location. So here you can see some spinal cord images with the white matter, and here you can see it's not very clear, but you have the butterfly of the gray matter in the spinal cord. So these data were acquired in 79 patients, including 27 female and 52 male, and the average age of our patients was 65 years old. So in my internship, I focused on the T2 star weighted and the magnetization transfer ratio metrics. So I will present you now the processing pipeline for T2 star weighted images. So the first step of the processing pipeline was to automatically segment the spinal cord thanks to the PropSec tool of the spinal cord toolbox. So once we obtained the spinal cord segmentation, we corrected them manually if it was needed. The second step was to register our patient's data into the PAM50 template from the spinal cord toolbox, thanks to the spinal cord segmentation we had. So this registration process enabled us to have each subject images in the same space so that we could produce some spinal cord maps. Then we applied the inverse deformation, so bring the PAM50 template into each patient's faces. And this inverse deformation enabled us to get the white matter and gray matter probabilistic atlas into each subject spaces so that we could, for each subject, identify some region of interest in our images. And thanks to this region of interest, we could then extract metrics, so get the mean value of intensity for a given region of interest at a given vertebral level. So our three regions of interest in the T2 star-weighted images were the whole spinal cord, the whole white matter, and the whole gray matter. So now for magnetization transfer ratio, it's almost the same processing steps. We just have one more step to compute the magnetization transfer ratio from the empty off and empty on images with this formula, but then we also have the spinal cord segmentation, the registration to the PAM50 template, and the inverse deformation. And we can see that here we have two more regions of interest, which are the dorsal column and the lateral funiculi. So now that we extracted our metrics, we used some multilinear regression model to analyze the variation in our metrics. So to do so, we used our metrics as a variable to explain. And we tried to explain this variable thanks to the UPDRS. So UPDRS is the Unified Parkinson Disease Rating Scale. So it's a scale that enables us to, that indicates how advanced Parkinson disease is in our patients. And we also use the age as a control variable. So here our parameter of interest is the beta parameter, which is the least square estimator of the causality link between our metrics and the UPDRS. So the interpretation of such a coefficient is that everything else being equal, an increase in UPDRS score of 1 point implies an increase of beta in the metric. So having that in mind, we computed, we used this regression model for each metric we extracted in each region of interest. So here in line, you have the vertebral level where we extracted the metrics, and in column, each region of interest being the spinal cord, the whole white matter, the whole gray matter, and the ratio between white matter and gray matter. So what we can see here is that for T2 star weighted images, we have two significant increase of the ratio between white matter and gray matter with respect to the UPDRS score at C2 to C4 level and C4 level. So when it comes to magnetization transfer ratio, we also have two significant results. So we did exactly the same thing, but with our two new regions of interest. And here we have one significant decrease of gray matter at level C2 to C4, and one significant increase of the ratio between white matter and gray matter at level C3. So in conclusion, what we saw is that quantitative MRI is sensitive to microstructural changes in patients with Parkinson disease. And we mostly saw some changes in gray matter compared to white matter. So for now, we don't really know how to interpret these results. It's possible that it's due to neurodegeneration, but we don't have the full interpretation for now. We also developed a processing pipeline for our data that is developed in order to be as replicable as possible. So our goal was really to limit the human biases during the process. So for instance, by using some automatic segmentation. And the project is not finished yet because we already have data for other metrics such as diffusion images, but this data needs to be processed and analyzed. And we also have data for healthy controls. So the next steps will be to analyze these next metrics and to compare our results to normative values in healthy controls. So thank you very much for your attention. I would like to thank all the participants to the study and the members who helped in the MRI acquisition for this project. Thank you very much.
Thank you, Camille, for this interesting talk. So Olivier, do we have any question on the chat?
Yeah, there's one question in the chat. So Victoria Madge asks, which UPDRS scores did you use? Is it only, for example, part 3, or did you use all 4 parts?
I think that in this project, we only used part 3, but we also divided the score in 2 parts because, as you have seen, we only had the images for the cervical levels. So we divided the upper part of the UPDRS score to see if we could have some more precise results. And in fact, in the results for magnetization transfer ratio, we didn't have any significant results for the total UPDRS score, but we had some significant one for the upper part of the UPDRS.
Okay, thank you. Another question from John Laporte, who asks, do you have a theory on why the ratio of white matter to gray matter was significant in a model, while neither white matter or gray matter by themselves were significant?
Yes, so I think it's because, well, the ratio between white matter and gray matter has been shown to be more robust to individual variation. And as you have, so we don't have many control on the individuality. We only introduce the age as a control variable because we don't have more variable to use. So maybe it's more significant for the ratio because it helps to be more robust to these individual factors.
Okay, thank you. These are all the questions in the chat.
Okay. Thank you very much.